Good day, everyone. For today's discussion, we are going to talk about another topic that is all about molecular evolution, wherein it is a field of study that explores the genetic changes and evolutionary processes that have shaped the diversity of the Earth. And this topic also focuses on the genetic material such as the DNA, the proteins, and this will help us to uncover the mechanism and the relationship of the molecular biology. So, by investigating how the molecules have evolved over time, the molecular evolution provides insights into the origin, adaptation, and the evolutionary relationship of the organism. And as we can recall from our previous discussions, we can say that cloning is a process of making an exact copy of an organism, since as the development of the scientific engineering continued to emerge and the process of producing an exact copy of an organism became possible. Thus, we can recall that cloning may occur naturally or it can be performed in a laboratory setup. Thus, when we say natural process of cloning, it can be explained by the twins. When a single fertilized egg splits into two embryos, these embryos then develop into individuals with nearly identical genetic makeup. And twins can be identical or monozygotic, meaning they share like 100% of their DNA, or fraternal dizygotic, meaning they share like only of 50% of their DNA like the regular siblings. When we say the two ways of when we say that there are two ways of cloning that can occur in a laboratory setup. These are the artificial embryo twinning and the somatic cell nuclear transfer. When we say artificial embryo twinning, it somehow uses the same approach to natural process, but it is being carried out in a petri dish instead of inside the mother's womb. So a very early embryo is separated into an individual cells, which are allowed to divide and develop for a short period of time inside the petri dish. These embryos are then placed into a surrogate mother where they finish developing. And since all of the embryos came from the same fertilized egg, that's why they are genetically identical. The second one is called the somatic cell nuclear transfer. And this kind of cloning undergoes several processes. The somatic, or the other cell containing a complete set of chromosome pairs, is isolated from a certain animal. And then this somatic cell is then cultured into the media that causes it to lose its identity as another cell. And the egg cell from the egg cell donor is isolated from a different animal. And the egg cell's nucleus containing the DNA is being removed, and the egg cell and the somatic cell are fused by an electric pulse, and then it is being implanted to a surrogate mother and to continue its development. And since the offspring receives all of the a copy of the chromosomes from the somatic cell, that's why it produced the exact copy from that certain organism. And those are the two commonly known cloning processes that occur in a laboratory setup. So much for that introduction about the cloning processes. We will explore more about the information about integration cloning and expression. And this discussion aims to discuss the history and discovery of the technique, its mechanism of action, or how does the technique being used, and some applications of the technique in terms of the medical and clinical field, and the modern or new interventions or adaptations of the discoveries for the technique. So first is all about the discovery of the technique. So before there's a specific technique that is being used in cloning nowadays, there is this like identification on how does the technique was first discovered, how does it being used, and how does it influence the scientific field. And so for the first discovery is the discovery of the DNA structure, wherein the elucidation of the double helical structure of DNA by James Watson and Francis Crick laid the foundation for understanding how the genetic information is stored and transmitted, wherein Watson and Crick published that the DNA is a double helix structure that twists to form the ladder-like structure where we think of when we picture what is DNA looks like. Thus, what we know about the DNA today can largely be credited to James Watson and Francis Crick, who discovered the structure of the DNA in the year 1953. Despite there being many important and contributing discoveries before and after their work. So this is like the year where they discovered the DNA's double helix or spiraling intertwined structure, which is fundamental to our current understanding of DNA as a whole. Like when they discovered that the DNA is a double helix strand, using their trials and error, Watson and Crick were able to assemble the DNA model that demonstrated that the DNA strands are anti-parallel, meaning the two strands of the DNA moves to opposite directions, and the DNA in a form of double helix were in the DNA strands of pair by a complementary base pairing, wherein the adenine is paired with thymine, and the cytosine is paired with guanine, and the outer edges of the base pairing is are being exposed, and this allows the access to replicative and transcriptional proteins. The second discovery of the technique is the development of the recombinant DNA technology. This recombinant DNA technology is also known as the genetic engineering that involves modifying the genetic makeup of an organism by introducing foreign DNA or by manipulation of the organism's own DNA. And this recombinant DNA technology technique also comes in several processes. First is the isolation of the genetic material. This first step is to extract the DNA from cells, and this is often done using specific enzymes that break down the cell walls and membranes, allowing the DNA to be collected. The second step is cutting the DNA at some specific locations. The restriction of enzymes or the molecular scissors are used to cut the DNA by specifically defined places. These enzymes recognize specific DNA sequences and cut the DNA at these sites. The next one is the amplification of gene interest. The specific segment of the DNA that contains the gene of interest is copied multiple times using a technique known as the polymerase chain reaction. And this by using this technique, this makes it easier to work with the gene as there are many copies of it. The next uh, the next process that is being involved in this technique is the insertion of the DNA fragments into a vector. The vector, the vector is often the plasmid, wherein the plasmid is like a small circular piece of the DNA, and it is also cut with the same restriction to enzymes, creating a complementary or the sticky ends to those on the gene of interest. And this gene is then inserted into a vector. And the sticky ends of the DNA and the vector combine and are sealed using the enzyme called the DNA ligase. The resulting plasmids contains the recombinant DNA. Next in the process is the transfer of recombinant DNA. The recombinant DNA molecule is then introduced into a host organism, commonly the bacteria or the yeast cells. And this process is known as the transformation. The host cells with the recombinant DNA are cultured. As they multiply, they replicate the recombinant DNA and express the foreign gene. And lastly, for the process in terms of the development of the recombinant DNA is the screening or selection. Finally, the cells that have successfully taken up the recombinant DNA are selected or screened using specific techniques. This might include growing the cells on selective media or inserting a marker gene at the same time as the gene of interest. And another thing about the development of the recombinant DNA technique is that it made it possible to study the sequence of the genes, to understand the inherited diseases, and to produce the gene-encoded medications such as the insulin and the growth hormone. Next discovery is the discovery of the plasmid. The discovery of the plasmid dates back to 1940s, and these plasmids were first identified in Staphylococcus aureus bacteria by Joshua Lederberg in 1952. And these plasmids became widely used as a cloning vectors due to their ability to replicate independently within the host cells. And this also refers to the unique qualities of plasmids that make them ideal tools for the genetic engineering. As an independent replication, the plasmids are like small circular pieces of the DNA that exist separately from the chromosomal DNA within the cell. They have their own origin of replication, a particular sequence of the DNA where the replication begins. And this allows them to reproduce independently of the cell's chromosomal DNA. Each time the cell divides, the plasmids also replicate, ensuring that each daughter cells receives a copy of the plasmid. This feature is particularly useful when it comes to cloning, since it allows for the rapid production of multiple copies of the desired genes. Also, as a cloning vectors in the field of genetic engineering, a vector is a DNA carrier that transfers genetic material from one cell to another. And due to the plasmid's small size and independent replication, plasmid serves as an efficient vector. And that is why the scientist can insert specific genes into plasmids, then introduce the plasmids into the cells. And once being taken up by the cells, these plasmids now carry the specific genes and replicate and express these genes. These plasmids are also fundamental in terms of protein production, because along with the inserted genes, the plasmids carry regulatory sequences that control the expression of the genes. This means that the host cells cannot only replicate the DNA that's had been inserted into the plasmid, but it can also use the inserted gene to make the protein it codes for. This capability has a significant application, such as the production of insulin and other drugs. Next discovery technique is the discovery of the expression system. The development of this system in relation to integration, cloning, and expression has been instrumental in enabling the efficient production of functional proteins and clone genes. The two early example of this technique are of such system rather are the bacterial expression system using the E. coli and the yeast expression system using the Saccharomyces cerevisiae. In terms of the bacterial expression system, the E. coli is commonly used bacterium in molecular biology and also in biotechnology. The researchers have developed the expression of system based on E. coli to drive the transcription and the translation of the clone genes into a functional proteins. In this system, the gene of interest is introduced into E. coli cells. The E. coli cells provide a favorable environment for the replication of the plasmid and expression of the gene, leading to a production of the desired protein for an organism. In terms of the yeast expression system, a type of yeast, or the Saccharomyces cerevisiae, is another popular host organism for the expression system. Yeast cells are advantageous for protein production because they have a well-established genetic and biochemical background. In the yeast expression system, the gene of interest is also introduced into a suitable plasmid vector, which is then transformed into the yeast cells, and these yeast cells carry out the replication of the plasmid and transcribe and translate the gene, resulting it in the production for the desired protein. And the development of this expression system have optimized various parameters to enhance the protein expression levels, such as the choice of promoters, enhancers, and the transcriptional regulators. This system is also have been engineered to facilitate the protein purification by incorporating affinity tags or fusion partners. The next technique that was being discovered is called the cloning of insulin gene. The cloning of the insulin gene, as an example of integration, cloning, and expression, revolutionized the production of the therapeutic proteins through recombinant DNA technology. The breakthrough achievement was made by Herbert Boyer and Stanley Cohen, who successfully expressed a recombinant insulin protein using an E. coli as the host organism. From what is seen in the image, the insertion of the recombinant DNA bacteria involves introducing the DNA molecule into the bacteria cell through transformation. And once inside the DNA can integrate into the bacterial chromosome, and recombinant DNA are copied, allowing for the replication on expression of the inserted DNA and the production for the desired protein.
Good morning. So let us proceed to number 6, which is the development of mammalian expression system. Researchers develop expression systems based on mammalian cells, allowing the expression of cloned genes in more complex organisms. These systems became crucial for producing recombinant proteins with proper post-translational modifications. So, the development of mammalian expression systems has been a significant advancement, particularly in the field of recombinant DNA technology and cloning. Mammalian cells are used because they can properly process and modify complex proteins, which is crucial for many therapeutic proteins and antibodies. In other words, the mammalian expression systems are like factories inside mammal cells that can make important proteins. Scientists use these systems because mammal cells, like those in our bodies, are good at making complex proteins the right way. The technique of cloning with mammalian cells involves putting a gene into a special carrier called a vector. This vector has instructions that tell the mammalian cell how to use the gene to make the protein. So when the vector is put into the mammalian cell, the cell reads the gene and make the protein. So this protein can then be collected and used for many things like making medicines or studying how proteins work in the body. Using mammalian expression systems and cloning techniques has made it much easier for scientists to produce important proteins that were hard to make before. An example is there is a scientist who wants to produce a drug to treat a certain disease. So, using the mammalian expression system, the scientist can clone the gene for this protein into a vector designed for mammalian cells. The vector contains the instruction that ensures that the gene is expressed in the mammalian cell. So, once the vector is introduced into mammalian cells, the cells begin to produce the protein. Because mammalian cells are good at modifying proteins, the resulting protein is correctly modified. So, this protein can use as a drug to treat the disease. So, as you can see, the mammalian expression system made it easier to produce proteins with the correct modifications. Seventh is the advancement in DNA sequencing. So, improvements in DNA sequencing technology facilitated the rapid identification and characterization of genes, accelerating the cloning and expression of novel genes. So, ang advancement in DNA sequencing have greatly influenced cloning and protein expression. Researchers can now accurately select and clone genes of interest, leading to more targeted studies and protein production. So, this precision in gene selection allows the researchers to study the function of that specific gene, investigate it, and even produce proteins for different kinds of purposes. So, these improvements have also streamlined cloning techniques, making gene insertion into vectors more precise and successful. As what I've mentioned earlier, vector contains the instruction that ensure that the gene is expressed in the cell. Understanding gene function has also been enhanced, aiding in the design of experiments for desired protein characteristics. DNA sequencing has also paved the way for personalized medicine by identifying genetic variations that impact disease and treatment responses. By sequencing the gene of an individual, scientists can identify genetic variations that may show signs of certain diseases, like how the DNA microarray works. This information would lead to the improvement of treatments to the specific patient, wherein the effectiveness of that treatment being done is high while minimizing the risk or side effects. So, the DNA sequencing has transformed cloning and protein expression by deepening our understanding of genes and enabling more precise and effective manipulation and expression technique. 8 is introduction of viral vectors. So viral vectors derived from viruses such as retroviruses and adenoviruses were developed for gene delivery and expression in mammalian cells. Viral vectors offered higher perfection, efficiency, and facilitated the study of gene function in vivo. So, viral vectors are modified viruses that can be used to deliver genes into the cell. So, kaning viral vector, tig-deliver ni siya. So, this is in vivo because this happens inside the body. So, 'di ba? This is derived from viruses such as retrovirus. A retrovirus is a type of virus that inserts a DNA copy of its RNA genome into the DNA of a host cell that it invades, thus changing the genome of that cell. In this case, the retrovirus is modified to form viral vectors in a way that its ability to replicate and cause diseases is removed. After that, the virus is then engineered to carry the therapeutic gene of interest, wherein once it infects the cell and delivers the gene to the DNA, it then begins to produce proteins encoded by the gene. So an example of this is forming vaccines. The viral vector delivers the gene encoding the antigens into host cells, wherein once the antigens are produced, an immune response is triggered, leading to the production of antibodies and activation of immune cells specific to the pathogen. Ninth is Genome Project. The completion of genome sequencing projects, such as the Human Genome Project, provides comprehensive maps of genetic information, enabling researchers to identify genes of interest for cloning and expression studies. So, the Genome Project, often referred to as the Human Genome Project, was an international scientific research project that aimed to sequence and map all the genes of the human genome. This project was completed in 2003. So, the primary goals of this project were to determine the sequence of chemical base pairs that make up human DNA, identify and map all the genes of the human genome, and develop technology for analyzing DNA. So, this resulted in the discovery of thousands of genes, providing researchers with a roadmap for cloning and gene expression investigation. So, with this, scientists could now easily access DNA sequences of genes, facilitating their cloning into vectors for further study or expression. Lastly, is the emergence of CRISPR Cas9 technology. So the development of CRISPR-Cas9 gene editing technology revolutionized molecular biology by allowing precise genome editing. So CRISPR-Cas9 has been integrated into cloning and expression workflow for gene knockout, knock-in, and other applications. So CRISPR-Cas9 allows scientists to edit genes with remarkable precision, enabling the insertion, deletion, or modification of specific genes in a genome. Kaya, 'di ba, it is an gene editing technology. So, researchers can now use this to precisely insert genes into vectors at specific locations in the genome. In gene expression studies, CRISPR-Cas9 has provided researchers with powerful tool to manipulate gene expression levels with high accuracy. So by activating or inhibiting the expression of specific genes, scientists can gain insights into gene function and regulation. This has opened up new possibilities for understanding and manipulating the genome, advancing both basic research and potential clinical applications. So, an example of this is when CRISPR Cas9 is used by researchers to study the function of a gene called SOX2 in mice. Kaning XOX2, it is known to play a crucial role in embryonic development, but its exact function in the adult brain was unclear. So, by using CRISPR-Cas9 to selectively disable the SOX2 gene in the brains of adult mice, researchers were able to observe its effect. They found out that SOX2 is essential for maintaining the stem cell population in the adult brain, highlighting its importance in brain function and regeneration. As you can see, the CRISPR-Cas9 is really essential in studying the function of specific genes as it is able to edit the gene precisely. So this can provide valuable insights into the roles of specific genes in various biological processes, advancing our understanding of genetics and potential therapeutic applications.
So cloning integration and expression are related, but distinct processes in molecular biology. Cloning integration involves inserting a foreign DNA into a vector for replication, while cloning expression additionally ensures the foreign gene is transcribed and translated to produce a functional protein. So the mechanism of action and how the technique work in cloning integration. The mechanism of action in cloning integration, it involves the incorporation of the foreign DNA, typically a DNA of interest, into the genome of a host organism. This process can occur through several mechanisms. So, homologous recombination. So, this mechanism involves the precise insertion of foreign DNA into a specific genomic locus by exploiting sequence homology between the foreign DNA and the host genome. Homologous recombination requires the presence of regions of sequence identity between the foreign DNA and the host genome, facilitating the exchange of genetic material. So during this process, the two double helices are close together and spool the double brand break and then naay mo attach nga MRX in which its complex functions primarily in initiating DNA double strand break repair by processing the broken DNA ends to facilitate strand invasion and subsequent recombination between homologous DNA molecules. This spool exits MRX to perform 5' to 3' resection. And here it comes the RAD51 strand invasion homologous recombination. The protein called REC-A or RAD51 in eukaryotes plays a crucial role. It binds to single-stranded DNA and promotes invasion of a homologous double-stranded DNA molecule. So this process is facilitated by the REC-A filament which forms on the single-stranded DNA, and then D loop or displacement loop is a structure formed during the homologous recombination where a single-stranded DNA segment invades a homologous double-stranded DNA molecule, displacing one of the original strands and forming a loop. So the two holiday junctions are crucial intermediates as they allow for the exchange of genetic material between the two DNA molecules. They can be resolved in different ways, leading to either the formation of crossover products in which it results in genetic recombination or no cross or non-crossover products where no genetic exchange occurs. The resolution of holiday junctions is tightly regulated and is essential for proper DNA repair and genetic diversity. So here's the video to better understand during the cloning integration. The second mechanism action is the non-homologous end joining. So this non-homologous end joining is a mechanism that joins two ends of DNA molecules together regardless of the sequence homology. So in cloning, the non-homologous end joining can be exploited to insert foreign DNA into the host genome by inducing double strand and breaks at specific genomic loci and relying on the cell's repair machinery to ligate the ends, sometimes resulting in the insertion of the foreign DNA. So the non-homologous end joining is a DNA repair process used by cells to repair double-strand breaks. It can be used in cloning integration to integrate foreign DNA fragments into a host genome. So non-homologous end joining involves recognizing broken DNA ends, binding them, processing them, and ligating them together. However, this process can result in small deletions or insertions due to imprecise repair. So here's the video for better understanding about this mechanism action.
To see how a cell repairs a double-strand break in DNA by non-homologous end joining, let's imagine that we have a parental DNA duplex containing the sequence A, B, C, D, E. We then imagine that the duplex is broken by a double-strand break in the C region. Degradation of regions from each side of the break, which are generally less than 10 base pairs long, may occur. This process is sometimes called resection. Q protein binds around the broken ends, leaving the actual DNA ends exposed. Q also recruits the DNA PK catalytic subunit, also called DNA PKCS. DNA PKCS recruits the nuclease Artemis and phosphorylates it. Artemis trims any single-stranded tails that are present at the break. Ligase 4, acting in a complex with XRCC4 and XLF-Cernanos, ligates the broken ends together. As a result, the duplex is rejoined, but note that any region that was removed by a nuclease will be missing in the repaired duplex. In this case, joining does not restore the DNA to its pre-break ABCDE sequence because the C region was removed by nuclease digestion. We see how a DNA double-strand break can be repaired without deletion or imprecise join in another video, how to repair a double-strand break by homology-dependent double-strand break repair.
Another mechanism action is transposition. Transposition involves the movement of DNA segments known as transposons or jumping genes from one location in the genome to another. Transposons contain sequences that encode the enzymes necessary for their movement, and this mobility can be harnessed in cloning to facilitate the insertion of the foreign DNA into the host genome. So here's the video for better understanding this.
A transposable element or transposon is a nucleic acid sequence in DNA that can change its position within a genome. For this reason, they are sometimes called jumping genes. This can sometimes result in the creation or reversing of mutations, as well as altering the cell's genetic identity and genome size. Transposons can be found in almost all organisms. There are two main classes of transposons. Class 1, also known as retrotransposons, is first copied in an RNA intermediate through transcription. Then that RNA is copied back into DNA using reverse transcription. The resulting DNA is then inserted back into the genome, making this a sort of DNA copy and paste. Class 2, on the other hand, has no RNA intermediate, and the element remains in DNA form throughout the process of transposition. An enzyme called transposase recognizes the DNA sequences at the 3' and 5' ends of the transposon and will cut the transposon out of the DNA and reinsert it somewhere else, making this process more like DNA cut and paste. If you have any questions, don't hesitate to ask me in the comments. Until next time.
So the last one is viral integration. Some cloning techniques involve the use of viral vectors to deliver foreign DNA into the host cells. Viruses have evolved mechanisms to integrate their genetic material into the host genome as part of their replication cycle. In cloning, viral vectors can be engineered to carry the gene of interest and deliver it into the host genome through viral integration.
When a retrovirus infects a host cell, the viral RNA genome is released into the cytoplasm where the viral enzyme, reverse transcriptase, copies the viral RNA into single-stranded DNA. The viral RNA degrades, and reverse transcriptase synthesizes the complementary second DNA strand. The double-stranded viral DNA enters the nucleus where it integrates into the host cell's DNA genome using the viral enzyme integrase. The integrated viral DNA is called a provirus.
The mechanism of cloning integration can also depend on the specific experimental design and the tools, techniques employed to manipulate the DNA of the host organisms.
Okay, let's move on to another techniques used in gene cloning, in which it's called expression system. So, what is expression system? So, to understand it more, let's tackle first what is expression system. Well, an expression system in biotechnology refers to the process used to generate a specific protein from a gene. It is a method that allows a gene to be expressed as a protein within a host organism. This system are used in research and industry to produce proteins for study or to manufacture products such as insulin or antibodies. So this report it provides a sufficient overview of the 6 fundamental steps that I was included that involve an expression system of gene cloning, a vital technique in biotechnology. So, these methods used or this method, the expression system, have 6 major steps. So, let's break down each steps for us to understand those steps. So, first steps, the isolation of gene of interest. The isolation of the gene of interest. So, these steps involves identifying and isolating the specific gene that you want to clone. These typically achieved using restriction enzymes, which are proteins that can cut DNA at specific sequence. This allow you to cut out the gene of interest from the rest of the DNA. So, I'll provide examples later after na mahuman na to-discuss ang 6 steps. So, next for the step 1, we proceed to step 2, which is the step 2 is its insertion into a vector in which the gene inserted in the vector often use a plasmid. So, what is plasmid? Well, plasmid is a small circular piece of DNA that separates from chromosomal DNA found in cells. Plasmids are most commonly found in bacteria, but they can also be found in other organisms such as yeast and plants. So, why plasmid is often used as a vector in an expression system? Well, it's because it's easy to handle and can replicate itself once inside the host cell, making many copies of the gene it carries. It also have makers that help identify cells that have successfully taken up that plasmid. These features make plasmid efficient tools for producing foreign genes into cells and producing proteins. So that's why in an expression system, often they when they're kanang undergo na sila vector or mangita sila vector nga usually they used plasmid because of that characteristic. Like easy to manipulate, self-replication, produce siya, and high protein production. Muna siya ang reason why are kanang usually they use plasmid as a vector. And to further go deeper, no, ang vector di ay is genetic vector is a DNA molecule used to carry a foreign gene into another cell where it can be replicated and are expressed. Mo na meaning sa vector. So that's why if they undergo sila ginana they use often plasmids or viruses inserted into a vector. So after you isolate gene interest, it is inserted once the gene of interest is isolated, it is inserted into a vector, which is typically a plasmid, a small circular piece of DNA found in bacteria. The vector acts as a vehicle carrying the gene into a foreign cell. This creates what is known as recombinant DNA. So, what is recombinant DNA? So, recombinant DNA or rDNA is artificial created DNA that combines sequence from 2 or more different organisms. Mauna siya ang recombinant DNA. Next, for step 3, we have transformation. After na ma-insert na katong isolation gene of interest, ang i-insert siya sa vector kay ang next step is to introduce the vector carrying the gene of interest into a suitable host organism. This process is known as transformation, in which as the bacteria are commonly used as host or organism because they are easy to grow and manipulate in lab setting. So after that, we move on for a step, the step 4, the selection. After transformation, the cells are allowed to grow. A marker gene is usually included in the plasmid, which allows for the selection of cells that have successfully taken up the plasmid. This marker gene usually confess resistant to a specific antibiotic. So, only the cells that have taken up the plasmid can grow in the presence of the antibiotic. So, in simpler terms, selection, or the step 4, the selection, is the step where we figure out which cells have successfully taken in the plasmid with the gene we're interested in. We do this by including a special marker gene in the plasmid that makes the cell resistant to a certain antibiotic. Then we grow these cells in the presence of that antibiotic. Only the cells that have the plasmid and those the antibiotic resistance will survive. So therefore, like, it will test in selection, dili magpili. Like in selection, di ba naman dyan ay certain a lot of candidates. So in anong mahitaboan ni, anang prosesa or anong steps kay katong tanan nga kanang tanan nga na ni undergo na sa 3 steps kay move siya sa 4 steps. So therefore, in ang tabo diri kay in selections, di ba, we have those qualifications. So the qualification manggod nga needed ani in order nga sila mo select is that kanang if kanang certain gene kay ang plasmid o ang kanang certain inserted gene and kay kanang cells nawa because cells that have the plasmid and those the antibiotic resistant will survive. So therefore sila mapilian kay sila may naka-survive along the way. Like naka-build in na jud ba, like ang katong vector o ang katong isolated gene of interest kay nakon gani na naka-build up na sila compatibility sa usat-usa. So therefore, like maka-form na sila o maka-form na sila of certain genes kay na compatible naman, like strong na ilang band nga na. So for the 5th steps, ika-5th steps kay expression. All of us knows about expression, the gene expression. Na makabaw naman na. So, pero like ang expression diri sa using expression system in gene cloning kay expression once inside the foreign cells, the gene of interest expressed, meaning the cell used the instruction provided by the gene to produce the corresponding protein. This process is often induced by specific substances that initiates the transcription of the gene, the first step in protein production. We all know about na jud atong transcription because transcription is the first process in gene expression. Gene expression has 2 processes. First is the transcription, next is translation. So, diri sa expression kay mo undergo siya og kanang transcription. Kay like, di ba na compatible na ang gene. So therefore, pwede na siya mo work, pwede na siya mo replicate or mo read sa DNA informations na carried out anang gene. Kaya compatible naman. So next is the protein purification. This is the last step. So the final step involves purifying the protein that has been produced by the foreign cells. This is necessary to ensure the purity and integrity of the protein for further study or use. This step usually involves a variety of techniques such as centrifugation and chromatography to separate the protein of interest from other cellular components. So, what is centrifugation? Centrifugation is the process used centrifugal force to separate a components of cell based on their size and density. Is often the first step in protein purification. Next, kay chromatography. This technique separates protein based on their properties such as size, charge, or affinity for a particular chemical. This mixture passed through a column, and different proteins move through the column at different rates, allowing them to be separated. So this is the several techniques used in protein purification. Why naman needed ang protein purification? Well, protein purification is necessary because it's needed to ensure that the protein of interest is isolated from other proteins and cell components. This process ensures the protein's purity and functionality, and it allows for a concentrated sample of the protein for further study use. So, dito siya purification of protein. Like as what I said a while ago, no, magbutang tag certain example nga, magbutang kog mag-say kong example nga ni-undergo sa 6 steps in the expression system. Ang giuse nga vector kay plasmid. So, isolating of gene of interest, the gene that codes for the protein we want to study. Mo na ang isolation of the gene of interest. And let's say, ang insulin ang ato ang kanang gene that codes na to for protein we want to study. So, insulin na itong gipili. So, insulin is identified and isolated from human DNA. So, manata sa step 1, i-isolate na nato ang insulin. So, let's move on for the step 2, insertion into a vector. The insulin gene is inserted into a plasmid. A plasmid is a small circular piece of DNA that separates from the bacterial chromosome. Therefore, diba, remember, ang step 1, ang insulin, ang ato nang isolated ang insulin sa human DNA. So, after ito ga-isolate ang insulin, ato na siya inserted sa plasmid. Kaya naman sa insertion into a vector, which is ang atong vector kaya ang plasmid. So, next is step 3, transformation. The plasmid now carrying the insulin gene is introduced into a bacterium like E. coli. This process is called transformation. Gi-carry ni plasmid si insulin gene niya. Iyang gi-introduce niya into a bacteria like E. coli. So that process is called transformation. And next is step 4, selection. The bacteria are then grow on a special medium. Only those bacteria that have successfully taken up the plasmid, and therefore the insulin gene will survive. So it means like compatible na si plasmid ug si gene naka-unison sila nga kung para, diba, how would you survive kung di ka compatible sa imong environment or sa imong gikuan, like sa imong pangingutang. Muna, it's like just like that po sa selection na during selection napabilin ang mga successful na ang gene of interest kanang insulin gene nga gi-carry siya sa plasmid na compatible lang duha, so therefore they were able to survive. Then next is step 5, the expression. As the bacteria grow and divide, they produce the insulin protein. This is the expression step. Last steps kay protein purification. The insulin protein is then extracted and purified from the bacteria for further use, such as treating diabetes in humans. Mao na ang nitabo. So therefore, mo na ang six major steps in expression system. So I'll provide another example for us to understand the 6 steps or the 6 major step in expression system. So, ang first is isolation of the gene of interest. So, ang ato na sang kanang gene nga interest nato kay kanang growth hormone. So, therefore, sa step 1, gi-isolate na nato ang growth hormone nga atong paon. Ang step 2 ka-inserted into a vector. So, This growth hormone gene is then inserted into a plasmid. Still, ang tong vector gamiton kay plasmid. A small circular piece of DNA found in bacteria. Yeah. Next is transformation. Third na ha. The plasmid now carrying the growth hormone gene is introduced into a bacterium gihapon nga such as E. coli. Mura gihapon nga bacterium. Then next is the selection. Kanang selection, the bacteria grow on a special medium. Only the bacteria that have sex will take up the plasmid, hence the growth hormone gene will survive. So, as what I have said repeatedly, selection is like kanang na certain qualification na dyan na. Like, if compatible na ba ang plasmid sa gene of interest nga gi-isolate. If di sila compatible, how would they survive after a long run? So, like, how would they survive into a long run if dili sila compatible? Sorry for my kanang what I've said a while ago. Sayop to siyang pagka-construct sa word. So, mana. Like, as what I have said, no? Like, selections, it had, as what I have said, no? Selections, it have some qualifications such as compatibility. If compatible na ba ang plasmid o ang gene of interest to survive? If na sila unity nga para maka-build up para sa ilang survival mode. Mo na it happen. Like, in this selection, ang mapilihan na dyan ka na compatible na. Next, the fifth steps is expression. As the bacteria grow and divide, they produce the growth hormone protein. And lastly, the protein purification. The growth hormone protein is then extracted and purified from the bacteria for further use, such as treating growth and disorder in humans. Treating. Next is, lastly, the protein purification. The growth hormone protein is then extracted and purified from the bacteria for further use, such as treating the growth disorders in humans. So,
let us now move on to the application of cloning integration and expression to the medical or clinical field. So before we discuss some applications to the medical or clinical field of cloning, let us first take note that cloning itself poses ethical considerations. So some of these applications is still on progress or still on early research because of that reason. So first in the list of application of cloning integration and expression to the medical and clinical field is the drug production. So drug production is done by recombinant DNA technology, in which involves this process is used to produce drugs and vaccines. The process involves inserting a human gene, such as one for insulin, into bacteria or yeast cells, which then produce the protein that the gene codes for. This protein can then be harvested, purified, and used as a medicine. This technology has been used to produce a variety of drugs, including insulin, growth hormones, clotting factors, and more. It has been also crucial for us, especially for the production of vaccines, especially at times in the pandemic where it is very in need with the production of vaccine. So next is gene therapy. So gene therapy involves replacing a faulty gene in a patient's cells with a healthy copy. How this is it done? So this is done by cloning the healthy gene and integrating it into a vector, usually a virus. So, what is the purpose of using a virus? It is because virus can infiltrate the human body. So, it can have high high resistance to the body's natural killers, such as, for example, your cells, immunoglobulins, and the the virus is already not harmful because it's already been modified to cure the gene of the patient's cells. Example of these therapies that had been approved for treatment for certain types are for cancer, genetic eye disorders, and spinal muscular atrophy. So next is cancer research and treatment. So cancer research has and treatment has genetic engineering can be used to modify immune cells to fight cancer. So one example of the immune cells are the T cells or CAR T cell therapy. So the T cell from a patient is immunified to express a receptor specific to a patient cancer cells. So this makes the cell CAR T cells able to detect the cancer cells that's in the body of the 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 person. So based on our previous learning, we have learned that that not all cancer cells can be detected by our immune immune system. Therefore, in this way, the cancer research and treatment devised a way for the immunoglobulins or the our antibodies to detect those cancer cells that is hard to detect. So next is gene editing. So one process or method that is used for gene editing is CRISPR-Cas9 as discussed earlier. So this technology is a revolutionary tool in gene editing and has been widely used in research to study genetic disorders and other diseases. It allows for precise modification of specific genes in a patient's cell. While it's extensively used in research, its application to the clinical practice is still in early stages due to concern about safety, ethics, and delivery methods. Moving on to stem cell research. This stem cell research is a well-established field, embryonic stem cells or ES cells in short, which have the potential to differentiate into various cell types, as used extensively in disease modeling, drug testing, and regenerative medicine. However, though this has widely used in research, the use of embryonic stem cell raises ethical concern, which has led to the development of induced pluripotent stem cell or iPSCs as an alternative. So why does the embryonic stem cell produces ethical concerns? It is because um, you in gain in gaining or acquiring embryonic stem cells, embryo must be formed first. So you are altering the development of an embryo or putting it into a stop because the stem cell will be used for other purposes. So it poses ethical consideration or ethical concerns to be um, performed. So in that concern, there are alternatives that are developed to solve the problem. So one alternative is the induced pluripotent stem cell. So instead of getting stem cells from embryonic stage, which are the stage where mature the process where first process where humans will be eventually formed to fetal and be born. So an induced pluripotent stem cell is like reversing the process bot instead of to bottom to bottom to top, you will start to top to bottom. So somatic cell will be reversed backward. If reverse backward, a somatic cell has already been specialized or differentiated by extraction. So it already has its specific function and purpose. So in this process, the induced pluripotent stem cell, the somatic cell will be reversed back to a stem cell. So it will go back in time and be and produce or transform as a stem cell. So this process is pioneered by Japanese researchers that is used instead of embryonic stem cell research. So next is the tissue engineering. Cloning and integration techniques are used in tissue engineering to create artificial tissues and organs. Cells are cloned and integrated to scaffolding, create functional tissues that can be used for transplantation or research purposes. This application is already been growing rapidly that involves creating functional tissues and organs in a lab. However, it is promising. The complexity of many tissues and organs means that this technology is mostly still in research and developmental stage. So this tissue engineering is still under process of development, and it's still acquiring information, trials, and many experimentation for it to officially be used. Based on our discussion of the application of cloning integration and expression to the medical and clinical field, we have known that technology or advanced technologies that take us further or one step further to solving our problems, especially in the medical or clinical field, in discovery of drugs, treating diseases, and discovering new techniques or technologies to solve our problems. However, it is also important to consider ethical considerations or concerns regarding about the use of these technologies, to examine the purpose and intent of the use of these technologies, as well as to set boundaries and limits to practitioners or to the people that will be using this technology.
So, here's the modern new intervention, adaptation, new discoveries on the technique, on the cloning integration and expression. So, first is optogenetics. So, what is optogenetics? So, optogenetics is a technique in neuroscience in which genes for light sensitive proteins are introduced into specific types of brain cells in order to monitor and control their activity precisely using light signals. So, optogenetics allows researchers to control how nerve cells communicate. And also in optogenetics, genes encoding for absence are introduced into the cells of interest, often through viral vectors or genetic engineering techniques. Once expressed, this absence can either excite or inhibit the activity of the cells when exposed to light. By using fiber optic cables or implanted devices to deliver light to the tissue, researchers can then precisely control the activity in these cells in real time. So optogenetics has revolutionized neuroscience research by enabling researchers to investigate the roles and the specific neural circuits in behavior cognition and diseases. So it has also shown promise in other fields, including cell biology, where it can be used to control activity of non-neuronal cells, and in bioengineering, where it has application in developments of novel therapeutic strategies. So second is multi-gene expression systems. So what is multi-gene expression systems? So it is the advances in synthetic biology that has led to the development of multi-gene expression systems, allowing for coordinated expression of multiple genes in various organisms. Multi-gene expression systems refer to the genetic constructs or platforms that are allowed for the coordinated expression of multiple genes within a cell or organisms. These systems are designed to enable the simultaneous production of proteins, often specific functionalities for complex biological processes. Last is the yeast and bacterial system. So, what is yeast and bacterial system? So, this is yeast and bacterial systems has been optimized for higher expression levels and increased stability of cloned genes. Yeast and bacterial refers to microbial organisms such as Saccharomyces cerevisiae or baker's yeast and Escherichia coli or E. coli, used in molecular biology and biotechnology for cloning, integration, and expression of genes. This system offers several advantages. Well characterized genes, genetics, arrays of cultivation, fast growth and high, reproduction rates, expression systems, post-translational modifications, industrial applications.
For the modern, new interventions, adaptations, new discoveries on the technique. By integrating CRISPR-Cas9 technology into cloning processes, researchers can precisely edit DNA sequences, improving the efficiency and accuracy of cloning experiments. This advancement enables targeted modifications, facilitating the creation of custom genetic constructs with greater precision. Next is high-throughput cloning platforms. The advanced automation and robotics enable the rapid processing of large volumes of DNA samples simultaneously, reducing labor and time requirements. These platforms streamline experimental workflows, allowing researchers to conduct high throughput screening and analysis, accelerating the pace of molecular biology research and biotechnological innovation. Using synthetic biology principles and tools allow researchers to design and construct custom genetic systems tailored to specific applications. By engineering genetic circuits and regulatory networks, scientists can optimize gene expression levels and protein production in host organisms, enhancing efficiency and enabling the development of novel biotechnological solutions. Next is directed evolution strategies. Directed evolution methodologies involve iterative rounds of mutation and selection to engineer proteins with desired properties. By subjecting protein libraries to evolutionary pressure, researchers can evolve proteins with enhanced functions such as increased stability, activity, or specificity. These strategies offer a powerful means of protein engineering for various biotechnological and therapeutic applications. Integrating cloning and expression techniques streamlines the process of producing recombinant proteins by combining DNA manipulation and protein expression into a single workflow. This integration has significant implications for various industries, including pharmaceuticals, where recombinant proteins are crucial for drug development and production. Additionally, in agriculture, the ability to express specific proteins in crops can enhance their traits, such as resistance to pests or tolerance to environmental stresses. Moreover, in research, the rapid and efficient production of recombinant proteins facilitates studies on protein structure, function, and interaction, advancing our understanding of biological systems. Looking forward, continued advancements in cloning and expression technologies hold promise for addressing current challenges and driving further innovations in fields such as in medicine. Continued advancement enable the development of personalized therapies and novel treatments tailored to individual patients' needs, leading to improved healthcare outcomes. In biomanufacturing, optimized production processes based on cloning and expression techniques enhance cost effectiveness and scalability, driving the development of biopharmaceuticals and industrial enzymes. In synthetic biology, engineered organisms with custom functionalities can be designed for various applications, ranging from biofuel production to environmental remediation, opening up new possibilities for addressing global challenges and improving quality of life.